Thank you for all of you coming, those here and those on the Zoom. I'm really happy to introduce you to Dr. Carolyn Smith, who's currently a postdoctoral research fellow at Duke University in the Department of Psychology and Neuroscience. Carolyn got her BS in neuroscience from the University of Massachusetts at Amherst, and then a master's and a PhD in behavioral neuroscience from Boston College. She then started her postdoctoral research with Dr. Stacy Bilbo at Harvard University, and after a couple of years, they both decided it was way too cold in Boston, so they migrated south to Duke University, where Carolyn and Dr. Bilbo have been since then, since about 2019. And Carolyn's been had a very productive training experiences, both as a graduate student and undergrad and a graduate student and as a postdoctoral fellow, enjoying all the environments of Research Triangle Park. She's been funded by an NIH F32 postdoctoral fellowship, and then recently, this year was awarded an NIH K99R00 Pathway to Independence Award, which is quite a coup. It's a very prestigious award. And she will allow her to transition from postdoc to a faculty position. She's published a number of research papers, reviews, commentaries, and scientific articles. Quite impressive, again, for a postdoc, over 35. Been involved in many scientific societies, and besides the Society for Neuroscience, she's been involved with behavioral neuroscience and psychoneuroimmunology. Say that 5 times fast. So you can see that she covers a lot of interesting areas that we're also quite excited about. She's received awards for her research and has been active also in teaching and in mentoring. So today she's going to tell us about her recent work on environmental toxicants and stress, on social deficits and how those deficits can be rescued by bugs and dopamine. Carolyn.
Thank you so much for the introduction, and thank you for having me here. I'm really excited to tell you guys about my research today, and I hope that I can give you a little bit of background on who I am as a scientist, my current project, and then a little bit on the future directions that I'm interested in exploring moving forward. So a key theme that has sort of run throughout all of my work is an interest in social behavior. And this is because social relationships and the ability to navigate complex social worlds is really critical to health and well-being throughout the animal kingdom. And deficits in social communication or social behavior are present in almost all neuropsychiatric diseases. And so because of this interest, I did my PhD, as Debbie was saying, at Boston College in the lab of Dr. Alexa Venema. And here we were really interested in understanding what are the neural mechanisms that underlie social behavior in the healthy brain. And we focused on the adolescent period because we know that adolescence in particular is a phase of life that's characterized by a heightened emphasis on social interactions with peers, as well as a lot of social novelty seeking and risk-taking behaviors. And in particular, we were interested in focusing on a couple of socially relevant systems in the brain, the oxytocin system, which is important for things like social behavior, social recognition, social reward, and the opioid system, which we typically think of in the context of drugs of abuse, but is also really important for mediating things like social pleasure and social pain. And obviously I don't have time to go through all my graduate work today, but just to give you a sense of what I was doing broadly, we used a combination of in vivo pharmacological manipulations using stereotaxic surgery and brain-wide receptor autoradiography mapping to show just really broadly that both oxytocin and opioid systems act in the brain to promote sociability during the adolescent period, and that oxytocin and opioid systems in the brain are really replete with both sex and age differences, particularly between this adolescent critical period and adulthood. And so my interest in sex and age differences sort of started here and has continued throughout my work. And so this training really gave me a broad sense of how are the circuits in the brain that support social behavior set up and how do they function in healthy animals. But for my postdoctoral work, I was really interested in understanding how do developmental insults sculpt the way these systems are set up during development. And so to do this work, I joined the lab of Dr. Stacy Bilbo at Duke University because she's really an expert in understanding how early life events shape brain development. And so my work has focused on how a few different exposures during this perinatal period influence the way that the brain is set up. So we've done a little bit of work on infectious agents and drugs of abuse, but my primary, and how they're related to social behavior. But my primary work has really focused on how environmental toxicants influence the perinatal period and the developing immune system to lead to changes in social behavior. And actually, I meant to say this before I started, but if anybody has questions during my talk, please feel free to ask them whenever they come up. Just to give this a little bit of a clinical perspective or relevance, we're really interested in autism spectrum disorder because we know that autism is primarily characterized by deficits in social communication and social behavior, as well as changes in repetitive behaviors. And we also know that autism has a strong sex bias and prevalence, with about 4 boys diagnosed for every 1 girl. We also know that both genes and environmental factors contribute to autism risk. So if we look at autism liability, about 60% of this risk can be accounted for from genetic sources, but this still leaves about 40% of autism risk that is attributed to environmental factors, really broadly defined. And the one that we've been really interested in is traffic-related air pollution. And the reason we were interested in this is that there's very strong epidemiological literature suggesting that mothers who live in areas with high levels of air pollution during pregnancy or the first year of life have an increased risk of having a child with autism. And this is true even if you account for demographic factors. When we're thinking about this, it's not as though every mother who's exposed to air pollution will go on to have a child with autism. And we also know that the populations that bear the greatest burden of environmental toxicant exposures also tend to be those that bear the greatest burden of psychosocial stressors. And so we hypothesized that there might be a model in which maternal exposure to stress during pregnancy might increase maternal immune activation in ways that might make moms more susceptible to environmental toxin exposures. So the central question driving our research here was, does prenatal exposure to air pollution and maternal stress alter social behavior in mice? And what might be the biological mechanisms underlying this? So just to give you a bit of background as to how we do this in our mouse model, we take C57 Black 6 mouse dams and we time mate them, and then we give them via intratracheal administration diesel exhaust particles that we get from the EPA through a collaboration there, or vehicle every 3 days throughout pregnancy. And then to simulate maternal stress, during the 3rd week of gestation from E13 to E18, we combine this with a maternal stress, a limited nesting and bedding paradigm. And in this model, mouse dams are placed on a wire mesh crate and given 2/3 of a cotton nestlet. And this is based on a limited nesting and bedding model that's typically used as a postnatal maternal stressor. However, in this case, we put them all back into normal cages on the eve before they give birth, so that the pups are born into an environment with plenty of nesting and bedding material. So we're really limiting this exposure to the prenatal period or developmental window. And then throughout most of the rest of my talk, I'm going to focus on outcomes in the offspring. So we can look at behavior, analysis of the gut and the gut microbiome, as well as neuroimmune endpoints. And so we have 2 groups in these experiments, our vehicle and control exposed animals, and then our diesel exhaust particle and maternal stress exposed animals. So it's sort of a double hit model. So the first thing we wanted to ask was, well, do we actually see social behavior deficits in offspring following these exposures? And so to ask this question, we used a sort of a classical 3-chambered sociability assay. And in this test, our subject animal is placed into an empty middle chamber, and this chamber has doors so that they can investigate any of these 3 chambers. And then restrained on one side of the assay is a novel conspecific, and then on the other is a novel object. And so what we expect to find in a healthy mouse is that they will prefer to spend time interacting with the social stimulus because they're social and they need to get social information that's very relevant for them. So what we find is that if we look at our vehicle control exposed male offspring, they really spend about 80% of their investigation time investigating the social stimulus. However, our male offspring prenatally exposed to the diesel exhaust and maternal stress really fail to show this social preference, suggesting that they may have an impairment in either their ability to process social information or their motivation to explore a social stimulus. Interestingly, however, if we look at our female offspring, they don't display this deficit, suggesting that this is really in line with the male bias that we see in autism. We can also test animals in another iteration of this test, which is testing sort of novelty preference. So here, both of the stimuli they're exposed to are social. However, one is novel and the other is familiar. And here, what we would expect healthy animals to do is to spend more time interacting with the novel conspecific, as this is one that should have important, socially relevant information that they need to gather. And again, what we see is that our vehicle control exposed males show a significant preference for the social stimulus, while our diesel exhaust maternal stress exposed males, if anything, slightly prefer the safer, more familiar stimulus. And here it seems like the females may be slightly less social novelty preferring, but again, it's not as striking a deficit. Yeah?
Are males and females untreated different? Is there a sex difference in the untreated?
Yeah, so we actually, so these are vehicle controls, so they are treated with something. There's no significant difference between the control groups. And I don't think, during adolescence at least, we don't typically see sex differences here, but that's a great question.
Are you measuring time spent, or what's your
Yes,
parameter?
exactly. So we score these for basically nose poking at these smaller chambers, and then we quantify the number of seconds that they spend investigating each stimulus. So this proportion is actually of investigation time.
Yeah, I know you mentioned adolescence, but what is the age? I'm going to guess here.
No, that's a great question. So we test them between P30 and P35. So that's like kind of right before the onset of like pubertal steroid hormones. That's a good question. I think another question that you might be wondering is, well, is this really because of the combined stressor? Could you get the same effect if you just expose them to the air pollution or the stress by itself? And so we wanted to make sure we checked for that. And so what we did is we had groups where we exposed them to only the stress alone or only the diesel exhaust particles alone. And what we found is that it was only in the combined group that we see this deficit in the males, really suggesting that you need the synergism of these two to kind of unmask these changes. So we see that there's robust social deficits only in male offspring following this combination of prenatal diesel exhaust and maternal stress exposure. But what might be the biological mechanisms that underlie this change in behavior? And so we turned our attention first towards the gut-brain axis. And so you can see here in this depiction, we have many microbiota that live within our gut. And there's a number of mechanisms by which they are capable of influencing brain function. So they can act on the vagus nerve, which signals directly to the brain. They can also produce metabolites such as short-chain fatty acids, which can then be released into circulation and impact the brain. And they can also influence the immune system to change the release of cytokines also into the circulation. And what was also really interesting to us is that actually people don't talk about this as much, but more than 50% of individuals with autism have comorbid GI dysfunction. And there's been some really promising clinical trials of microbiota transfer therapy in autism, showing that it improves not only GI symptoms, but also behavioral symptoms for quite a long time. I think they've now moved as far as 2 years later. So this is really promising to us. And we also know that mouse models of like germ-free mice, which lack gut microbes or microbes entirely, have really robust deficits in their social behavior. So to do this, we collected fecal contents from offspring following these prenatal exposures. And then we can do 16S RNA sequencing. And for those of you who aren't familiar with this technique, I'll just very briefly tell you that basically there's this 16S RNA gene in bacteria that has this hypervariable region. And different species of bacteria have different variations within this region of the gene. So what you can do is essentially amplify only that gene, barcode the samples from each mouse, pull them together, and then sequence them. And what you can do is actually figure out what bacterial species are likely present based on the number of breeds that have each of these variations. And this is the most widely used method, I think. It doesn't give you as much information as something like metagenomic sequencing would, but it's usually used as a good first pass to get a sense of what's different. And so if we look at measures of diversity, and I'm only showing the data here for the male offspring during adolescence, what we find is that if we look at alpha diversity, which is basically a measure of how diverse or even is the microbiome within an individual animal. So a very even microbiome would be one in which each bacterial species is represented in the same abundance, where a very uneven microbiome would be one in which you say have a predominance of only one species of bacteria. What we actually found, somewhat surprisingly, was that our male offspring prenatally exposed to diesel exhaust and internal stress actually have an increase in the evenness of their gut microbiome as compared to vehicle control. We can also look at beta diversity, which is a measure of how disparate are the microbiomes between animals. And here we see there's a significant difference where our vehicle control males in blue really cluster nicely together, while our diesel exhaust internal stress exposed males are really much more disparate. And importantly, we looked in females and we didn't see any changes in females, again, in line with this very striking male bias. We have also since looked later in development and found the same changes at a later developmental time point. So these seem to be pretty robust findings.
Are you co-housing the controls on the trees?
So that's a great question, because housing has a huge impact on the microbiome. So what we did is we only use, like, each of these dots represent mice from different litters that were never housed together. So we have pretty good distribution of litters. But everybody starts out, I mean, what's kind of interesting is that the dams that will go on to be mothers in the vehicle control condition or the diesel stress start out as sisters. So we take like litter mates and then divide them into the treatments, which serves as a really good like housing control for housing differences in the microbiome because their baseline is all the same. That's a good question. Of course, the next question is, are there specific taxa of bacteria that are differentially expressed? Because these overall patterns are interesting, but harder to target and be specific. And so if we look at the genus level, it can be sometimes difficult to get differences at the species level. We see that both Bacteroides and Parabacteroides are significantly decreased in our diesel stress exposed males as compared to vehicle control. And this was really interesting to us because these are actually both species, or genera of bacteria, I should say, that are decreased in human patients with autism. We also wanted to look at the intestine because, of course, if you see changes in the gut microbiome, it's interesting to ask whether or not you might have changes within the gut itself. And so to do this, we used H&E staining of histological sections through the ileum, so small intestine. And we can measure things in these sections like the gut wall thickness, crypt length, and villi length within the intestine. And what we actually found was that there was no difference in gut wall thickness or in crypt length by treatment, but we saw a main effect of treatment if we look at villi length, which actually with longer gut villi in our diesel stress exposed offspring, and actually this was the case in both males and females, which was kind of interesting to us, and we're not exactly sure what it means. So certainly females are not exempt from any effects of these exposures. And there's some interesting work showing that actually villi length can change with nutrient availability, so longer villi are often seen in nutrient deficiency, and that was sort of an interesting observation to us. Another important aspect of gut function that we were interested in was looking at the tight junctions between enterocytes within the gut. And so there are these proteins that really mediate these tight junctions and keep the gut barrier tight so that bacteria are unable to translocate. And certainly in autism, there's discussion of the leaky gut hypothesis where things from the gut are more capable of getting into circulation like bacteria or bacterial metabolites. And so to assess this, we use gene expression of the ileum again to look at mRNA for occludin and zonryl occludins 1, which are tight junction proteins. And here we see that they're decreased in male offspring following exposure to diesel exhaust and maternal stress, and actually increased in the females. So a really sex-specific difference. And it's interesting to speculate, although it's purely speculative, is when we see some of these opposite changes in females, that there's some protective advantage that they infer or give to the females. The next question we wanted to ask was, well, given that we see these changes in the gut microbiome and changes in social behavior, is there a causal relationship between the two? So could we intervene at the level of the microbiome and rescue social function? And so to ask this question, we used a cross-fostering procedure at birth, because cross-fostering of offspring to a new novel mother on the day of birth has been shown to shift the gut microbiome of offspring to be more similar to that of the foster mother as opposed to the birth mother. And so what our procedure looked like is that we had pups that were either exposed to the vehicle control prenatally or the diesel exhaust maternal stress prenatally. And then on the day of birth, so within a few hours of birth, we fostered them to either mothers of the same treatment in these two conditions or mothers of the opposite treatment. So everybody is fostered in this procedure. And then the key question we're really interested in asking is, does cross-fostering to a vehicle control mother at birth prevent these social deficits? We also wanted to check and make sure that actually we see a reversal of the changes in the gut microbiome. It seemed like an important thing to check for. And so what you can see here is that in the dark blue dots are our vehicle-to-vehicle fostered animals, so our vehicle control group. And then our red dots are our diesel-to-diesel. And you can see that they're pretty disparate from each other. But the pink dots, which are the diesel fostered to the vehicle, again cluster more closely to the vehicle control group, and these are significant differences. So this cross-fostering to a vehicle control mother does appear to be normalizing the distribution of the gut microbiome. If we look at alpha diversity, we actually see here that again our vehicle control offspring are different from the diesel, although in this case it's actually going in the opposite direction, which surprised us, but they are all cross-fostered, and this is something we can talk about more if you guys have questions. But again, the diesel to vehicle are back up at vehicle levels. So we see significant shifts within the gut microbiome. So now if we look at our behavior, we again come back to our 3-chambered social preference test, and here I'm showing the diesel stress pups. Those fostered to a diesel stress mother really don't show a preference for social stimulus, as we saw before. However, those that are fostered on the day of birth to a vehicle control foster mother, again, see a restoration of their preference for a social stimulus. And here I'm also showing raw seconds spent in social investigation, and you can see that this is really driven by an increase in the number of seconds that they spend investigating a social stimulus. Interestingly, however, we actually did not see a difference in the vehicle control pups between the vehicle control and the diesel stress foster. So it's not as though we can induce social deficits by fostering them on the day of birth, which to me suggests that the microbiome is capable of rescuing behavior, but it doesn't represent the whole of the etiology underlying the changes. And I'll again come back to this later in the talk. Another thing that you might be thinking is, well, you're fostering them to a new mother on the day of birth, so what if your changes are just driven by a change in the maternal care that they're receiving, because now they have a mother that was not exposed to these environmental exposures. And so we really wanted to account for this, and so we pretty carefully quantified maternal behavior during the first 3 days of life. So here we have a histogram just showing we basically did 10 observations 3 times a day for the first 3 days of life. So during 3 different hours, and each of these is an observation. So anything in the warmer colors is active maternal behavior, and anything in the darker colors is when the moms were not on the nest taking care of the pups. And what we found was that actually diesel stress pups do actually receive a little bit more care than vehicle control pups, so moms spend slightly more time on the nest and more time looking and grooming, but it doesn't depend on whether or not they were raised by a vehicle mother or a diesel mother, suggesting that there's something about the pups that the moms can tell that they need a little bit of extra attention, but it's not what's behind this rescue of behavior in the vehicle control foster condition. So just to summarize what I've shown you so far, we find that our diesel exhaust maternal stress exposure is leading to social deficits, changes in the gut microbiome, and in the intestinal epithelium in male offspring only. And we can rescue these social and microbial changes via cross-fostering at birth. Yeah?
So question. So sometimes when you use new stressors into the system, the microbiome changes to adapt to that. And so when people look at the metabolic products, metabolomics, the change in the frequencies of the bacteria actually kind of adaptive, so you maintain the same level of metabolites. Did you look at any of the metabolic?
Such a good question, and I think this is a big, like the big downside of using this sort of the genus level characterization that we've done is that you wouldn't pick up, just like you're saying, you could have compensation there where you actually lead to no change in what the bacteria are producing because they can just change their compositions. We haven't done metagenomics yet or metabolomics, but it's something we're really interested in doing, and part of my K99 is to add that to this so that we could actually see, you know, if we have these changes in Bacteroides and Parabacteroides, are there pathways that are differentially up or down regulated? Yeah. Oh, sorry.
Did you look to see whether there are changes in cortisol?
Yes, so the moms have changes in cortisol during pregnancy, and the offspring on postnatal day 1 have increased cort, but it's gone by P8, which is interesting. So it doesn't seem to be sustained, but there is sort of a cort response to these exposures early in development.
I'm not sure if I missed it in the first summary, but were there any differences in, litter health, meaning size, growth trajectories, male to female sex ratios, anything else like that could be just a retardation of development in the males that were exposed? That's
such a good question. And I actually this morning I was like, I should have included that slide in here because that's a great question. So we didn't see any difference in litter size, no difference in male-female sex ratio within the litters, and we also tracked maternal weight gain during pregnancy pretty carefully, and we didn't see any differences there. So for the most part, they really seem to be behaving pretty normally.
And the pup weight and their growth, their weight gain over...
So the one thing I will say is we didn't see any changes in pup body weight at P14, P24, and then I think that's as old as we measured them, but there were no differences. But in our cross-fostering experiment, we did actually pick up at P35, diesel stress pups, regardless of who they were fostered to, were slightly smaller. So I do think there's something going on there, and we were thinking, we see this, like, they have longer gut villi, and so we were thinking maybe they're, slightly smaller because they have some nutrient absorption deficits or something. So it's something, it's a great question, it's something we're interested in.
And you think you would have seen that same behavior in the non-fostering experiment if you'd taken it out longer?
Yeah, that's a really good question. I think we've been trying to think a lot. One thing with the cross-fostering is that because they actually are delivered vaginally, so they do acquire microbes from their birth mother, and then they're fostered. So they actually get a microbiome it ends up being very similar to the foster mother, but they get this first inoculation with the birth mother's microbes. So we're interested in how different are these cross-fostered animals from the normal ones, because they get this extra microbiome sort of dosing.
Maybe I missed it. I know you showed the offspring with the cross-fostering, but do you actually know that the microbiome of the dams at parturition is different?
Can I come back to that at the end? Because it's actually more interesting than you would think. So I will definitely answer that question. Any other questions? Yeah.
So cesarean section changes, doesn't allow vaginal inoculation with the microbiome, right? So there's plenty of evidence that cesarean birth alters social behaviors and cognition levels. So have you considered anything like that with this model of cesarean as well as exposure to
I think that's a great point, and actually part of why we decided to take the cross-fostering approach that we did, because the ways you typically interfere with the microbiome would be to deliver by a C-section and then cross-foster so that they never get microbes from a birth mother, or to do like an antibiotic treatment or a germ-free mouse. The problem is that all three of those have changes in social behavior and also microglia, which I'm going to talk about in a minute. So we kind of ended up choosing to go for the cross-fostering because it seemed like it was the most natural and would be the least likely to disrupt all of those processes. But that's a really interesting question. And something I'm interested in is how do these birth modes impact the microbiome, colonization, and then social behavior. Yeah, for sure. Okay. So the next question we wanted to ask was, what neural changes might underlie these social behavior deficits? And so we decided to focus on the mesolimbic reward system within the brain. And this is really comprised of ventral tegmental area neurons that produce dopamine that project to a variety of brain regions, including, but most largely, the nucleus accumbens, which is a really critical brain region for social reward. And this has been shown to be disrupted in human patients with autism, as well as in animal models of social behavior. And a variety of neuropeptides and socially relevant molecules act within the nucleus accumbens, including oxytocin, opioids, and dopamine systems to mediate social behavior. And so we were interested in seeing whether or not these systems would be disrupted by these prenatal exposures. And so to do this, we started by doing tissue punches in the nucleus accumbens and looking at RNA, and then we've also done some immunohistochemistry. And I will just say, given my graduate work in oxytocin and opioids, I was really interested in whether we might see changes there. And to my surprise, there was absolutely no difference in either of these systems, as kind of always happens. But when we looked at the dopamine system, here we saw some very striking differences. So on the left, we have dopamine D1 receptor mRNA, and we see that this is decreased in male offspring following our diesel exhaust maternal stress exposure, and in the females, it's actually increased. And to see whether or not the change in receptor might be coupled with a change in the fibers coming into the brain region, we used immunohistochemistry to look at tyrosine hydroxylase fiber density. Tyrosine hydroxylase is a dopamine synthesis enzyme, and so we use this to label for dopamine fibers. And here again, we see a decrease in the male offspring and a slight increase in the female offspring following diesel exhaust and maternal stress exposure. And this is also during adolescence, I should say, when we looked at the behavior. So given that we see these decreases in both receptors and fibers for the dopamine system within the incumbents during this time period, we wanted to ask, could we activate the dopamine system and rescue behavior that way? And so to do this, we used a chemogenetic approach using DREADS. And for those of you who might not be familiar with DREADS, DREADS, this acronym, stands for Designer Receptors Exclusively Activated by Designer Drugs. So it's essentially inducing a system in the brain that you can then activate when you want to activate it. And the way that this works is that you would use a pre-dependent viruses to transfect specific neurons with either an excitatory version of this receptor or a control virus, which just encodes this mCherry. And this virus, the excitatory receptor, is a G protein coupled receptor that should be inserted to any cell membrane of cells that have this pre-construct. And then by activating it with your designer drug, which you can apply later, you can actually basically drive action potentials in this specific neuron subset that you're interested in. So we took dopamine transporter 1 pre-mice, which should have pre only in dopamine neurons, and we injected it locally into the ventral tegmental area. And so this allows us to basically transfect inject these viruses locally into the VTA and transfect only these dopamine neurons in this brain region. And then what we can do is right before behavior, we can inject peripherally our designer drug, which is clozapine N oxide. And this is a compound that, for the most part, has no other effects on brain function other than to activate these receptors very potently. And this is just an image showing tyrosine hydroxylase in the green and mCherry in the red, showing that we can successfully transfect this neural population within the ventral tegmental area. And so when we do this manipulation in our mice, here we have the data for sociability. So all of these mice received the clozapine N oxide to activate the neurons, but in these two groups, they only were injected with the control virus, so nothing should happen. And indeed, if we look at our vehicle control infected males, we see that they show a preference for social stimulus, as we would expect. And if we look at our diesel exposed males, they don't show a preference, so recapitulating our previous findings. However, if we activate the dopamine system, we see that this rescues their social behavior phenotype. And the same thing can be seen if we look at seconds spent in investigation. So we have a significant difference between social and object investigation time in our control mice, and then no difference between the two in our diesel stress animals, and then a rescue of this by driving the activity of these dopamine neurons within the VTA. So this suggests to us that we can activate the dopamine system and rescue these social behavior deficits. So the final thing we wanted to ask in the brain was, well, what's leading to this change in the dopamine system in the first place? And we had recently done some work in the lab showing that microglia, the resident immune macrophages of the brain, actually play a critical role in eliminating dopamine receptors in the nucleus accumbens during adolescent development, and that this has important implications for social behavior. So we wanted to ask, do we see changes in microglia in our model? And we also wanted to ask this because we know that microglia respond very strongly to signals from the gut microbiome. So germ-free mice, which lack microbiota, have very hyper-ramified microglia, and this is particularly true in males. And so in these cells, they're macrophages that live permanently in the brain, And their ramification state is typically thought of as an important indicator of their function. So you can see a control microglia looks like this. It has these branches, and these germ-free mice have these really hyper-ramified cells. And the thought is that when they become really amoeboid and bring in their processes, they're often producing a lot of pro-inflammatory cytokines and are traditionally thought of as being activated, while when they're more branched, they're more surveillant of their environment, and they're actually constantly extending these processes and kind of feeling what's in their surroundings. And so if we look at our diesel exhaust maternal stress exposed animals, we actually see a very similar hyperramification phenotype in the male offspring, but no change in the females, suggesting that there may be similar hyperramification here. Just to get a final sense on a little bit more granularity of what might be different in microglia, we did some gene sequencing to understand what their gene expression profiles might look like. And so to do this, we isolated microglia from the nucleus accumbens using a CD11B magnetic bead column pull-down method to isolate just this one cell type from this brain region. And then we did next-generation RNA sequencing to look at their gene expression. And I'm not going to have time to go into all the details, but I just want to show you the kind of overall patterns that emerged here. So if we look just at these overall in the males and the females, these are volcano plots. So this represents genes that were more highly expressed in our diesel stress males, and these are genes that were more highly expressed in the vehicle control, and if it's above this line in red, it means it was significant. And you can just see immediately that many more genes were differentially expressed in the males as compared to the females. And we've started to delve into what these genes that are upregulating by following diesel exhaust and maternal stress are, and actually a number of them come up in cell proliferation and even some cancer signaling pathways, which is not my area of expertise, but we're trying to understand what it means for these cells and what their function might be. So just to put all of these things together, what we find is that our diesel exhaust and maternal stress exposure leads to changes in social behavior, changes in the gut microbiome, and intestinal epithelium, and changes in microglia and the dopamine system, in male offspring only. And we have shown that we can also rescue these changes in social behavior in the gut microbiome via cross-fostering at birth, or by activating the dopamine system, temporarily before the behavior during adolescence. And this suggests that either of these might represent points at which you could intervene to rescue function. And I think personally that the microbiome is a particularly attractive candidate to explore as a therapeutic target, because it's so much easier to intervene at that level than it would be to go into the brain and try to activate the dopamine system, although both are really interesting. However, there's still some outstanding questions and things we're trying to answer. And one big one would be, well, how does cross-fostering at birth restore the gut microbiome and social behavior? Because, the first thing you would think of would be, well, the diesel exhaust and maternal stress is actually changing mom's gut microbiome because these exposures are happening directly to her. And then that is then leading to transmission of this altered gut microbiome to the offspring at birth. And that was kind of the hypothesis that we went into this with, and expected that this is what we would find. However, we were really surprised to see that actually there was no change in the maternal gut microbiome, the vaginal microbiome, or actually even the milk microbiome of moms. And so this is a plot showing mom's microbiome. And what you can see is we took samples at baseline, so before they were mated, and then every 3 days throughout pregnancy. And what you can see is the different colors represent the different days of throughout gestation. And those do cluster differently, so that they're separate from each other. So the microbiome is changing over the course of pregnancy. But the diesel moms and the vehicle moms, which are these circles and the Bs, they're really intermingled at each time point. So we really couldn't pull out any differences between the different treatments in mom's microbiome during pregnancy. And I didn't show the data here for the vaginal or milk microbiome, but we did have to learn how to milk mice to get this data. So if anybody is interested in that,
Did you ever look at the lung?
So this is such a great question. We're interested in, we haven't, and I know some folks here are interested in the lung microbiome, which is something that I think is really neat and I think hard to study. So I'm curious to hear about that more. But these are direct diesel installations in mom to the lung, and so we're interested in getting more information about what's changing locally in the lung that's then leading to some systemic change in the lung. And kind of on that note, thinking, okay, so if it's not microbes via one of these methods, what might it be? And we do know that if we look at maternal blood serum at embryonic day 17.5, so right before they give birth, we see higher levels of TNF-alpha and IL-17A in mom's circulation. And so we're wondering if, since the cross-fostering has a rescue effect and it's happening at birth, perhaps there are differences in cytokines in maternal milk of the foster mothers that might be causing some of these changes in behavior. I'm also really interested in better understanding how the infant gut might be shaped during development, during gestation in this pro-inflammatory environment that might then change which microbes are able to live there. So thinking about it from maybe the microbes aren't the causal mechanism themselves, but a change in response to a change in the gut, which I think would be really interesting. Does anybody have any questions on anything else at this point? Yeah.
First of all, I'm sorry for kind of...
Yeah, no, this is great. I'm so glad you asked.
Cutting to what was really cool data here. So I think your point is not just on the cytokines, but even the composition of the milk might be different, as far as like lipid enrichment or other nutrients. And then that's leading to some of the adaptive changes potentially in the gut as well. So I think kind of maybe not just looking just narrowly at cytokines, but maybe more at the nutritional status of the milk that the pups are receiving through metabolomics or something might be kind of another way to look at it.
Yeah, absolutely. We would really like to do that. So that's actually something we have lots of milk that we would like to use for that. Also because we saw this change in villi length, and since we have increased villi length, we thought maybe they're nutrient deficient.
Like less amino acids or a shift in the change of lipids and stuff.
Yeah, and what's interesting is that like doing these assays with milk has proven to be challenging, and not a lot of people are used to working with milk samples, or they're very high fat content, so like even spinning them and getting like a even homogenization is like really challenging. So we've been trying to work through that, but we have a lot of it, and we would love to do that. Yeah, it's a great thing.
Have you looked at any of the exosomes that are produced by milk, since that's a big field right now, and if it's different between these animals that are the diesel versus the diesel?
We haven't, but exosomes are really interesting, and I don't know much about them other than that they sound really cool, and I wish I did know more about them, but we haven't looked at that, but that's a great thought.
Okay.
Yeah. Yeah.
So what are your thoughts on the sex differences, the females and female? Why do you have a weaker sex than males?
So that's part of why I think that the something intrinsic to the gut or to the animal itself must be driving some of these, because it is kind of surprising, right, that these male and female offspring get the same microbes, the same, everything, and only the male offspring are displaying any of these changes. And that's where I think it has to be some interaction with either the immune system or the microglia or the gut of the offspring that the microbes then interact with to lead to those changes.
If you feminize the male offspring or masculinize the females, what would you predict to happen?
So that's a great question. So I think I would predict that if you could masculinize females, you could induce some of the same susceptibilities. I think the timing is a little bit tricky, so our exposures are prenatal, and like the testosterone surge is happening around the day of birth. So, you have to think about when you intervened. But that is not to say that sex steroid hormones still have an impact in utero, right? Like placental location, if females are close to female fetuses, it can have an impact on masculinization in the female. So yeah, I think certainly steroid hormones probably confer some of the probably a large portion of the vulnerability that we see in males.
I think we can look at endocrine disruptors as well.
Yeah, absolutely.
So I'm going to ask the other side of the question then. If you feminize the males, does it become protective? And if you're going to go down that pathway, do we think that the effect of environmental estrogens, which seem to be more and more plentiful, is actually protective as far as, does all the extra Zyrnols and popcorn that we eat protect us from the diesel exhaust that we've been breathing all week?
Yeah, that's a great question. I mean, certainly, like, I like to think about these interacting exposures, which can be hard sometimes to layer on, but I think there's certainly the potential for that. And I think, I mean, male babies tend to be more vulnerable to, like, so many of these really early life exposures, and then females have this later window of vulnerability during adolescence, and so I think it's really interesting to think about the timing and why that is. Certainly I will say we do see changes in the females in some of these things, so it's also interesting to think about, like, why do they see changes on some aspects and often in very different ways from the males but not in others.
You know how long those changes persist? You look at adolescents, what about adults, 2 months, 3 months?
So certainly we've looked at the changes in the microbiome and in the gut and behavior, and those extend into adulthood. So they seem to be very persistent, despite the fact that it's, you know, yeah, they seem to hang around for a long time. I will say, I didn't mention this, but we also looked at pro-inflammatory cytokines within the gut just to see whether or not, like, is there just overt inflammation all the time, and we haven't seen any changes in pro-inflammatory cytokines in the gut throughout development, which is interesting. So it's not that they're maintaining just like huge inflammation persistently.
I know your expertise is in social behavior, but have you looked at some of those other behavioral aspects, like the memory and the risk-taking, and any sex differences between any sex differences in those aspects?
Yeah, that's a great question. So I haven't done it, but another student in the lab was looking at ultrasonic vocalizations, so like a measure of social communication early in development, and she has seen changes in these social vocalizations at like postnatal to 8, so early in development. So that is another metric. We have not looked at social like recognition, but we should look at social memory. The novelty familiarity kind of gets them a little bit, because if they have deficits, it could be because they can't remember that one is novel and one is familiar, but it's not really a full characterization. We did look at repetitive behaviors and anxiety behaviors, and we didn't see any differences there. So it does seem to be somewhat specific to the social domain.
The water maze test, some of those classical kind of working memory maze kinds that includes.
Yeah, we should definitely do that. We have not, but that would be a really good idea. Yeah.
Your beta diversity slide, where you have the exposed males are kind of all over the place. Could you correlate their beta diversity to the behavioral outcomes? Because you had a lot of variation in the behavioral outcomes. So is there maybe do a regression correlation analysis or something to see if like ones that were over here in the beta diversity actually was that, you know, get what I'm saying?
We should definitely do that, because we actually have microbiome samples from the same animals as behaviors, so I actually don't know why we haven't done that. That's a great idea. We'll definitely do that.
Can you comment on the number of mother litters that are being used in behaviors, since the animals are frequently between 8 and 10?
Yes, so we never take more than 2 pups from the same litter. So we use many litters. I can't think of the number for an exact experiment, but we really try to make sure that we distributed them across litters. That's a great point.
Can you comment about the hyperramification of the microglia? Can you hear me?
Yes, I think I can. Yes.
Thank you. Can you comment about the hyperramification of the microglia in the male mice? What age does that begin? What's the length of the lead there?
So that's a great question. So in germ-free animals, I think they have changes in microglia starting quite early in development, but whether or not you have more or less or hyper-ramified or not actually really depends on the brain region you look at. So there isn't really one pattern that's consistent throughout the brain. We haven't looked at the ramification phenotype at earlier developmental time points, but we do see changes. Another student in the lab has a paper on bioarchives where she's looked at microglial phagocytosis of glutamate synapses in cortex, and that's different as early as postnatal day 7. So this does occur, changes in microglial function occur pretty early in development. I hope that starts to answer it.
There were some questions on the chat. I think, Linda, if you can ask them, that would be great. Chat.
Can you all hear me?
Yes.
Yes.
Okay. I think there's just one question that wasn't asked yet. Isn't any, might have asked this question this is from Duzon John. For microglia, so big difference between GF and SDF mice. GF pup are conventionalized at different days. What their microglia expect to look like?
I'm not sure I understand the question.
Maybe if they're online, if they could unmute and ask the question themselves.
Yeah, you can unmute now yourself. That was the problem before.
Yes.
So my question is, so if we conventional germ-free mice at different age, so how do you think this affect the microglia development?
Oh, that's a good question. So there has been, you have like germ-free mice are reared in these incubators and they have no exposure to microbes at all until whatever day. Usually you take them out and you take them out of them and they're exposed to microbes, so conventionalized. And you can do this at different developmental windows. I do know that germ-free mice have very disrupted microglia and very disrupted social behavior. So I think, I'm not sure if you take them out later in life, I wonder whether microglia completely normalize in their structure, what would happen. I'm not entirely sure of the answer to that question, but I would assume that when you take them out would make a big difference for what happened, what the implications of that are for microglial function.
Yeah. One more question. So, if I remember correctly, when you in the exposed males that have changes, you actually saw an increase in the diversity of the microbiome. Is that correct?
Yes.
Yeah. So during that period, the immune system is also starting to develop, and the microbiome plays a major development of the immune system. So you think the changes you're seeing in the microglia are just cytokine mediated because there's more tolerance, there's more bacteria?
Yeah, that's a really interesting question. I think that would be really cool. I mean, we know that this period when we see this adolescent period where we have this increase in beta diversity is right after it's a couple weeks after they're weaned. And so their microbiome goes through this huge change. And so we were thinking that perhaps if you see this increase in diversity there, that's sort of different bacteria species are trying to fight to acquire a hold and to sort of figure out who's going to be present later. But I think it's a good point that it could be influencing microglia as well.
The microglia are contracting, right? Which means they're less.
They're actually getting more bushy. They're getting bigger. They're getting hyper-ramified. Okay. But certainly, and we actually do see changes in microglia early in development as well. But we're really interested in this question of how does the gut microbiome influence microglial function? Is it via cytokines? Certainly, microglia express other receptors for, say short-chain fatty acids, receptors like FAR2, FAR3. So we're interested in what might be the signaling pathways by which the gut influences microglia.
Can I ask a question in that regard?
Yeah.
Yeah, great talk, really fascinating. Have you tried to restore males providing Bacteroides and Parabacteroides isolated from the same mice, the control mice?
That is a great question and one of our future directions for research. So that's part of my K99 is to see if can we rescue microbiome changes, not by rescuing the whole microbiome, but just by isolating the specific strains of Bacteroides that are present in our animals in the vehicle control, and then giving them back to the diesel stress. So absolutely, yes. Yeah.
Hi, great talk, first of all. Thank you so much. Can you comment a little bit on how behavior could be the primary driver of these microbiome changes, right? Restrictive, repetitive behaviors in humans might cause different dietary changes, and how you kind of control that out of your experiments?
Yeah, and I think so I don't know that I think I cannot say we've controlled it out of our experiments. I think it's a really interesting point. I think another thing is that they could have social anxiety, right? And so it could be that being housed with other animals makes them more stressed, or that social interactions could be more stressful for them, which is why they avoid them. And certainly we know that stress can influence the immune system. It might influence what bacteria are capable of living there. So you could have a bi-directional relationship by which their behavior then, helps to maintain these stable changes in the microbiome. I think that's a really cool idea. Was there a question over here?
So overall, should pregnant women stay away from driving on the New Jersey Turnpike?
Probably, I mean, I don't know, I don't feel confident from my mice models to say anything about human epidemiology, but certainly it is, the literature, I mean, it's, you know, there's always some studies finding each thing, but it does seem like there are so many, I mean, there's so many exposures during pregnancy, but high concentration of diesel exhaust does seem to be, at least to some degree, related to risk, for sure. Yeah.
Do either the particles from the diesel exhaust or chemicals absorbed onto the particles make their way to the fetal brain?
So there are certainly like aerohydrocarbon receptors on microglia and in the brain, and I feel like there's been, you guys probably know this better than me, but there's been some papers showing in humans, I think, that particles can actually get through the placenta and into the developing fetus. So I would definitely believe that that's possible. And certainly, like, when you collect lung tissue from these animals, even weeks later, you see particle accumulation in the lung.
In the babies.
In the moms. And in the offspring, I haven't seen, like, to the naked eye, anything like that. But I don't think it's impossible by any means.
And then with that parameter, though, is there like changes in maternal oxygenation status or anything like that that could also be having an in utero effect?
We have not looked, but I think that's a great idea. It's definitely possible. Yeah.
Are you planning to conduct any dose response studies in order to establish the threshold of diesel exhaust exposure that can lead to such appreciable effects on social behavior?
That's a great question. I think this model has been in the lab for a while, and then they did do some sort of dose response to look and see which concentrations were both ecologically relevant and effective. But in the context of social behavior, we definitely haven't done that. So that's a great point.
And since your model showed that you need both the driving on the turnpike and taking away mom's mattress at the same time, how do you do a dose response in terms of two variables simultaneously?
Yeah, it's a great question. I know the model, the limited nesting embedding model, when people first started using it, they literally were like, is it effective if they have half a nestlet or a quarter of a nestlet or 2/3 of a nestlet? And so there was this like funny, it's like, and it seemed like having 2/3 of a nestlet was the right amount. But yeah, it's really complicated to figure out how, if you have these two. I think this is always the trouble with studying. Like, of course, these things are multifactorial, but when you're trying to model multiple things, it gets complicated very fast. Oh, yeah.
So I just want to clarify, you said that the microglia get more ramified with, okay, so how do you think that ramification and like pro-surveillance process is potentially leading to social deficits? Because I've always thought, I did microglia research in undergrad, and we always hypothesized that the overactivation and amoeboid morphology leads to that pro-inflammatory state that can then cause damage throughout development.
That's a great question. Yeah, so I think we know a lot more about what it means for a microglia to be amoeboid and making more cytokines, and like certainly if you had a traumatic brain injury or an infection, like that's what's going to happen. But I think what we've been really interested in lately is this idea, like we used to think that they were just holding their processes out there and they weren't doing anything, and now we know that they're actually, you know, surveying neurons, they're touching neurons. There's all this funny stuff about how they're stroking them or something, and then but they're also phagocytosing synaptic elements and eating things really actively, especially during these critical periods. So one thing we're interested in is, you know, they're normally supposed to phagocytose synapses during adolescence. Maybe they're phagocytosing the wrong synapses or too many synapses, or, you know, eating things that they're not supposed to when they're, or what are these contacts, what do they mean? And I don't think we really know the answer to that. That's something really interested in understanding.
Thank you very much.
I won't go through these because we don't have some introductions, but I did just want to say my thank you slide to my lab mates. I have wonderful undergraduate students who helped me with much of this work, and all my amazing lab mates and my advisor. So I cannot leave you without having this thank you slide up there.
I like the social distance.
This was our COVID lab photo. Okay. Yeah, thank you so much.